A very big hello to my wonderful PU2 champions. I hope all of you are doing super great. Well, this video is for those of you who are going to have their FA2 Biology FA2 examination really soon. And those of you are wondering, ma'am, where should I start from? I have no idea. Or there might be some group of children who might be thinking, ma'am, what are the topics I should not miss for topping the exam? So anyway, this video is going to help all of you. Just make sure you try to solve each and every question. Do not do rectification, okay? Do not by heart the answers. Rather, understand the concepts. The questions can be formed in different way as well. But I can guarantee you that these are the questions or basically concepts that are going to be repeated 101% sure. Ready? So are we ready? I hope we are ready. Well, extra benefits that you're going to get from here is that you might get some charts like this. So this chart is for the chapter Human Health and Diseases, my dear children. Over here, I have made the list of all the diseases and I have put the names of all the organisms that causes. Now, one very important thing, wherever you see mosquito, that means mosquito is a vector there. Vector as in someone who carries the protozoan here, someone who carries the virus here, someone who carries the helminthic worms, eggs over here or something like that over here. So they are carriers. They will be carrying the organism that actually causes the disease. Okay. So please know the name of the mosquitoes as well. That can be a question. For female Anopheles mosquito causes what? We all know, ma'am, it is malaria. Wacha, tell me then, what is a disease? You are given option and you are being told that spot the disease which is being spread by mosquito. And in that, malaria is not there, dengue is not there, chikungunya is not there, but filariasis is there. But you think filariasis is caused by this organism. You have no idea that it is spread by mosquito. You will lose your marks. So you should know that it is spread by mosquito. The name of the mosquito is Culex. What is its name? It is Culex. Now over here we know it is Anopheles mosquito, and over here both of them are spread through Aedes mosquito. All of them are females. Females are the one who drink blood. I'm not talking about human beings. Human beings also do not do that. Do not say anything bad about female, okay? Anyways, over here females do that for basically feeding their young ones out there. Okay, the larvae, they need blood meal. That's the blood meal. That's the thing. So yes, so Anopheles, Aedes as well as your Culex mosquito is there. Remember that Anopheles mosquito is a carrier or what do you call as a vector. Apart from that, it is also a host because life cycle of Plasmodia happens partially in the mosquito's body and partially in our body. So we and mosquitoes are the hosts as well for malarial parasite. Okay, so these type of two questions that can come will be sorted out from what do you call this particular page. Go through it properly. If at all you want it, what do you have to do? You have to follow us on our WhatsApp channel. You'll get the PDFs there. Okay, now human health and diseases, these are the questions that you have to definitely go through. Life cycle of malarial parasite. Malaria as a whole is super important. You should know in what stage that the malarial parasite get into our body. It is sporozoite stage. Is gametocytes formed inside our human body? Yes, gametocyte is formed inside the human body. Does sexual reproduction of the plasmodium happens inside the human body? No, it happens in mosquito's body. So those of you who are thinking like, oh my god, what is ma'am saying? If at all you're confused about the concepts, one short video is coming. Okay, or might have already been put. Watch it. It is going to help you out. Now, what is innate immunity? The difference between innate and acquired can be asked, and also the barriers of innate immunity. There are 4 of them. What do you call physical, physiological, cellular, and cytokine barriers. All of them, please know them. Differentiate between active and passive immunity. This is also a question. They can ask you. Basically, a man is bit by what do you call? Not mosquito, snake. And they are being given anti-venom. What does this anti-venom contain? What type of immunization is this? So anti-venom contains antibodies against this, preformed antibodies, and this is a type of passive immunization. Your body is not involving over here. Rather, you are being given ready-made antibodies. So this is passive immunization. Questions like that can be framed. So that's what I'm telling you. Whenever you have questions like this, if at all you understand the concepts, you will be able to solve more questions related to this. Got it? Now, what is allergy? Mention the symptoms of allergic reaction. Name any 2 drugs that would quickly reduce the symptoms of allergy. Allergy is because of secretion, that is histamine secretion. Now, you give antihistamines or you give steroids. In that way, it can be treated. Now, in allergy, what cells are involved? Please know it is mast cells that are involved. Ma'am, what does mast cell secrete? It secretes histamine and serotonin. histamine and serotonin. Ma'am, which antibody is involved over here? It is IgE, allergy, IgE. Allergy, IgE, tell that always, you'll remember it. How many antibodies do you have? GAMED. The first antibody to travel from mother to child is G. It travels through the placenta. The second antibody that travels from the mother's milk to the child is what? Is A. So you can remember A for Amma. Okay, so please remember all the small things out there. Yes. Now, what are lymphoid organs? Write the functions of the following in response. So you should know what are primary lymphoid organs. These two are primary lymphoid organs, where basically the lymphocytes are made, they mature and they become antigen sensitive. That means they know that I have to fight against antigens, but they do not fight over here. Remember, and they become antigen sensitive. They are sensitive to antigens. They don't do the action over here. So always remember, primary lymphoid organs are where things become antigen sensitive. What are effector cells? Effector cells are found in the spleen, lymph nodes and things like that. So basically, these T and B lymphocytes, when they reach the secondary lymphoid organs such as spleen and lymph nodes, my dear children, they are going to fight with the antigens, and that's when we call these cells as effector cells. So effector cells in the secondary lymphoid organs and antigen sensitive cells in the primary lymphoid organs. There are only two. Please know the functions of all of them. Spleen, lymph node, and there are more. There is MALT, M-A-L-T. Mucosa associated lymphoid tissue that occupies 50% of the lymphoid tissues out there. In MALT, under MALT comes your tonsils and your Peyer's patch. So mucosa associated, mucosa is the innermost layer. So whatever things are associated with this layer comes to be a part of what you call the lymphoid tissue. So your Peyer's patch is found in the mucosal layer of small intestine. That is why it is a part, Peyer's patch, okay. Peyer's patch. And what? And this tonsils is also a part. Cool. So please remember all of this thing. They can ask you, give you an option that which of the lymphoid organs are the one where the B and T cells become effector cells. So you should know it is spleen, lymph nodes, Peyer's patch, tonsils, or if at all MALT is written, then MALT. Cool. Yes. Cool. Now, with the help of schematic representation, draw on how infected cell can survive while viruses are replicated and released. So basically, over here, please understand, you need to draw the diagram of retrovirus HIV affecting, okay, HIV affecting a normal cell, and in that you should know the flow. HIV virus comes, sends its RNA first, RNA undergoes reverse transcription with the help of what enzyme? The name of the enzyme is super important, reverse transcriptase. Reverse transcriptase, okay, and then basically now it has formed the DNA gets into our host genome, and now it uses our machinery, forms its own RNA, forms its own protein, and now the whole virus is there. So you should know the flow. Please go through it as I am telling you. If at all you have any doubt, watch the Gunshot video, even if you feel that it's long. But my dear children, if that lengthy video can give you the whole marks, then why are you worried about spending your time out there? And I am telling you with guarantee, you will understand all the concepts. Okay. Now, how explain different techniques which help in cancer detection and diagnosis. So you have to talk about different techniques, and if they are asking you that which is the one that helps in obtaining 3D structure of the internal organs, it is CT scan. CT scan is for 3D structure of internal organs. Now, ma'am, I want to examine living tissue. MRI is there for examining living tissue. Please know that. Okay, so all these small things. Now, what are carcinogens? That means cancer causing agents. List any two type of carcinogens. So you should know physical, chemical, biological carcinogens are there. The most important ones are biological carcinogens. Okay, biological ones are the most important ones, such as oncogenes. In our cell, we have some proto-oncogenes which basically produces proteins and stuff like that. But if at all they are getting mutated, they make the cell cancerous. So we have proto-oncogenes within us. If they get mutated, they can make the cell cancerous. Apart from that, there are some viruses which have oncogenes and they transfer it to us, and then our cells also become what you call cancerous. So please know about biological things. They are super important. And list all the drugs and their side effects. All the drugs, my dear children, are super important. There is no way that I can give you a clue that what drug might be coming in. And I have a picture though which might definitely help you. And what are the effects of anabolic anabolic steroids on males and females. It is given towards the last part of your chapter. You should know that in females it leads to masculinization, mood swings, depression, all of this thing. Know all the symptoms. You might get MCQ questions based on that. Okay, so these are the things. Please remember one more thing that I have missed somehow over here. Example of autoimmune disorder. Example of autoimmune disorder. What is the example of autoimmune disorder? Rheumatoid arthritis. Rheumatoid arthritis. So please go through all these things out there. Okay, and here, as I said, I have put on all the drugs. This picture can be asked, and you will be asked for which drug is it or from where do you get. So this is the structure of morphine, and morphine comes under opioid. So this looks like a O. So O, opioids. Where do you get it from? Opium poppy, that is papa, where somniferum. What does it affect? The central nervous system and the gastrointestinal tract. What is heroin? Heroin is a depressant, slows down your body function. Okay, what is the other name for heroin? It is super important. Please remember, smack is the other name for heroin. And what is the other name for heroin apart from smack? It is diacetyl morphine, because you get heroin after acetylation of morphine, which is a drug that doctors give after a surgery. It is morphine because it is a sedative and a painkiller. Please know all these small details. Where do you get this from? Opioid from the latex, okay, from the liquid of opium poppy. Now cannabinoids, same thing. You have been given so many options. Marijuana, hashish, charas, all come under this only. It affects our cardiovascular system. Only the only one which affects the cardiovascular system. And please remember, if at all you are asked, what is a drug that is abused by sports people? It is cannabinoids. It is given in the NCERT. It is cannabinoids. Now if at all you see this type of thing, okay, this type of thing, please understand you're talking about a cannabinoid. This is a picture of cannabis sativa. Okay, now you have Coca alkaloids. Again, where do you get it from? A question can be coming in. What is a plant that is native to South America and gives you drug? That is erythroxylum coca. That gives you what? Cocaine. Cocaine is also known as coke or crack. CC. Do not worry. Okay, and where does it affect? Again, your central nervous system, just like how heroin and morphine were doing. But over here, they are affecting the dopamine transmission, which is why you will get a sense of euphoria and also hallucination. Well, there are two more other plants that can give you hallucination, and that can be also asked. What are the other two plants? Atropa belladonna and datura. Atropa belladonna and datura, they can also lead to what? Hallucination. And over here you have the picture of datura. Please identify it. So you can be given what is a plant that does not cause hallucination. So these two plants, and erythroxylum coca is given. Do not take them. Take the other one which is not there included. Okay, now other drugs, barbiturates, amphetamines, benzodiazepines, BAB. It helps in fighting depression as well as insomnia. That means if at all you're not able to sleep, this drug will help you. Do not take it. Now nicotine is responsible for increasing your blood pressure. Do not forget it. That is a drug that increases the blood pressure. Cannabinoid affects your cardiovascular system, but nicotine is the one that affects your blood pressure because it releases adrenaline and noradrenaline, things like that. Now the next chapter is microbes in human welfare. My dear children, all of it, you can get it in the gunshot video that I am talking about. I have explained them in detail, so you need not worry. I might be going up in bit speed over here because this video is just for question list. So actually I should not be explaining, but I thought I should. That's why I'm doing it. I'm so sorry, I have a habit like that. So name the products you get from distillation such as rum, brandy, whiskey, and without distillation such as wine and beer. You should know that. Okay, I have put the names over here, but you should know that. Now, name the microbes that help in the production of the following bioactive agents. These are the bioactive agents: cyclosporinase, streptokinase, statins. You have to tell from where and where you get it. Okay, again, a very important question could be coming in. It's found in 3 model question papers. Define the following: flocks and BOD. Super important. Please go through it. Now, describe the biological treatment of primary effluent. I'll tell you one thing. Go on, study the sewage treatment fully. They can ask you what is primary treatment. They can ask you what is secondary treatment. Any question can be coming in. So please, it's better to know what is sewage treatment as a whole. Now, draw a label diagram of a typical biogas plant. This can be asked. What is biogas made up of? Please understand it is made up of CH4, that is methane, not CH3, CH4. Apart from that, H2S, hydrogen sulfide, and also CO2. If in the option it is not H2S but H2, do not think it's wrong. It's also correct. Okay, it is also correct. But biogas is not made up of methyl alone, and always remember it is made by anaerobic bacteria, the ones that does not need oxygen. Now explain the role of microbes in household products. So household products, industrial products, biocontrol agents, all of them. And biofertilizers is given in NCERT. I know there are so many names, but we have to learn it. And one question that is associated with this is role of fungi as biofertilizers. So you have to talk about the fungi. Over here I have given them. So it's basically all the microbes as biofertilizers. If at all they are asking you what is a nitrogen fixing bacteria, remember cyanobacteria is also bacteria, this is also bacteria, but they are asking you about this bacteria, Rhizobium, Azospirillum, Azotobacter. Okay, if they are asking you what are the cyanobacteria that helps in fixation, then you should be talking about Anabaena, Nostoc and Oscillatoria. Okay, and mycorrhizal fungi basically provides what and all. Fungi provides basically phosphorus, that is very important thing. It provides phosphorus from the soil, it provides resistance to pathogen, tolerance to salinity and drought, and also helps in overall growth and development. It does not help in yield increase. It does not help in yield increase. That can be a question. What is the function of fungi? So you should know what are the function. So small details might be there, please go through it. Same thing, microbes in industrial products, you might be asked to do match the following question. Please go through it. All of them are super important. This is the first antibiotic that we got, penicillin. You got it from Penicillium notatum. It's a fungus. Okay, similarly, Aspergillus niger is also fungus. Yeast is also fungus. Rest of them, my dear children, this is also fungus. Rest of them could be bacteria. So please go through that. Okay. Household products, all the things super important. Go through it. Okay, I have given you over here so that if at all you access the PDF, at least you read through the options. Microbes as biocontrol agents, all of them are biocontrol agents. Please go through them. Now, biotechnology principles and processes, my dear children, these are the questions that you have. Describe the steps involved in recombinant DNA technology, all of them. Explain the method of naming of restriction enzymes by giving an example. And you should know the site of EcoR1 restriction site, that is 5GAATTC3-. Okay, so explain the steps involved in separation, isolation of DNA. Please know how is cell wall broken of a fungi, of a plant, everything. Then how do you separate it? With the help of chilled ethanol, my dear children. And later on you run it through electrophoresis chamber. And how do you make it visible? With the help of ethidium bromide. It gives an orangish red color. Now list out the steps of PCR technique, super important, can be given as a whole question or might be given in the form of MCQ. What is insertional inactivation? Give example of LAG-Z and also in PBR 322. Now what are the methods to introduce alien DNA into the host? How to make the cells competent? That question can be asked. Please know there is heat shot treatment, this and that and all of it. Now calcium treatment method. Now list any 3 tools used in recombinant DNA technology. Describe the structure of PBR 322. Please know where is the restriction enzyme site and everything. Super important from this chapter. Draw a neat label diagram of simple stirred bioreactor. Now they can ask you what are the vectors used for plants. It is Agrobacterium tumefaciens. Okay, what is a vector used for animals? It is always a retrovirus. It is always a disarmed retrovirus. So all of it please know. And apart from that, one more thing, plasmid. What are the features of plasmid? That is also super important. What are the features of plasmid? Cool. And so what is not can be asked in the MCQ question. Do not get it wrong. Now biotechnology and its application, again these many questions are there. List out the use of genetically modified plants. This is there in the model question paper. If you want virus-free plants, then what part of the plant will you use? You will use shoot and root apical meristem. Now with that I remember, please know what is soma clones and how it is different from somatic hybrids. And how it is different from somatic hybrids. Okay. Now one of the applications of biotechnology, this is important, okay, is to get pest resistant plant. Justify the statement with reference to BT cotton. So please know the BT cotton gene and how does it work, the CRY1, CRY2, what do they do, all of it. Explain RNA interference in plants and write a note on gene therapy. Please go ahead, know the steps involved. Lymphocytes are taken, they are being cultured and then this and that, everything. Draw a neatly labeled diagram showing pro-insulin and mature form of insulin. This is also important. What is biopiracy? Explain it with reference to basmati rice. Okay. And transgenic animals provide innumerable benefits to human beings. Explain the functions of transgenic animals. That can be also asked. Is there any question? No. So these are the few very important things. Go through each and every question. Take out time for that. And as I said, I am here, I have made a video where I have not went like this, like Shabdi Express, explain slow, you can watch that. And if you think that this video was helpful, what do you have to do? Click on the like button right now and share it with your friends as well, my dear children. And not just that, subscribe to the channel. Yes, if at all you think it was helpful. Yes, so bye-bye, take care, see you soon.